faculty. So today she will tell us about a comprehensive approach to detecting and correcting spurious transcriptome inference due to the RNA-seq read misalignment. Thank you and welcome.
Thank you for the wonderful introduction. Actually, I often find myself like have too much things to do and then never find time to to get things done. So it does help me to build up my confidence when you say that I accomplish a lot. Thank you so much for that comment. But it is true that I have been trying my best, and I always thought that maybe it just took me longer to finish certain things than other people. So I always find myself busy and then commit myself to something, but then it always takes longer to finish. So because I came from the background of doing data mining many years ago and then continue to do that, and nowadays like everybody moved to big data, and then I'm just one of them in the crowd. And we found that actually it's a quite fascinating area to work in, and I can really use my past experience in doing data mining and then to help in this field. In my group, we have quite a wide spectrum of a project. Actually, it's often led by graduate student, and each one of them take a different direction. So we end up with a bag of interesting project. Today, I'm going to share with you one of these. During throughout the day, actually, I met quite a few people, and then have a very interesting conversation and learned. And then also, I mentioned a few times that I do have other project very different from this one. But if you're interested, I can share with you later. For gene Caesar, so how we start to came that? So a few years back, and when I was at UNC, we were part of an NIH center. At that time, the center was doing the overarching theme is to using laboratory mice to study depression. So in that project, we have really many samples of different mouse strain, and then we want to, in this case, we want to do a differential gene expression analysis. Right? It's a relatively small project within that overall scope. So at the beginning, we thought, well, we just do microarray. And then later, we realized that maybe RNA-seq is a better way to do it. So we run RNA-seq. And at that time, it was our first time to actually look at RNA-seq data. It's a lot more. It's much richer than microarray data. But then we actually learned many lessons in a hard way, I would say. So, and then that's how this project came along. At the beginning, we said, well, maybe we just download the recommended analytics pipeline and by Illumina, and then we can just get the result. And then, because at that time when we get the data, it's about a month to the time we need to give a demo to or give a talk to an NIH program officer. So we're under the pressure, and then we just try to get some reasonable result fast. But then we were really surprised in a negative way, and then we have to come up with some solution. So for RNA-seq analysis, this is the standard pipeline. Some of you may already know this, in your heart. So you start with read, right? Read basically just short segment fragment, right? Come from somewhere from the transcriptome. And then because you don't know where they came from, when you just got the read from maybe the sequencing center, and then a typical way is like, let's do alignment, right? Either to the reference genome or to the reference transcriptome. So once, in this case, in this drawing here, let's assume we do the alignment to the reference genome. And then now so hopefully, most of your read, a high percentage of your read can be aligned like this. And then the next step is to call another, algorithm to do the assembly. So, and to basically tell you that which transcript is expressed and then by how much. Okay, that's a typical pipeline. And if this thing is done right, and then we hope that it can give us quite accurate estimate of gene expression level, and also it will help us to detect novel transcripts that people didn't know about. This is less noisy, than the microarray data. It's much higher resolution. But as we all know, RNA-seq data is much larger than a typical microarray data, right? It's obviously come with a new challenge that we have to face, and quite a few. So, as a standard pipeline recommended by Illumina at that time, TopHat is one of the commonly used alignment algorithm, right? And the Cufflink is one of the commonly used the assembly algorithm to enforce the transcript level. So basically, the output of TopHat would be the input of the Cufflink, right? And then, so this is exactly what we did at the beginning. Once we get the data, and then we run this. We download them, install them, and run them, okay? We saw that, okay, great. And then because our goal at that time was to do differential gene expression analysis, right? After we run this for every sample, we just see, well, which gene or transcript is highly differentially expressed. So, but it was not as easy as we thought, because all of our top-ranked transcript, differentially expressed genes, if you call, are pseudogenes, are annotated pseudogenes. We have to go down to the list very deep in order to hit a real gene somewhere. So, and in that case, one of our goals actually in the project was to study parent of origin. Okay, and then we did find something, but the effect is exactly opposite to what other people report in their paper. So, it's quite controversial, that piece of result. We can't just show that to a program officer and then without a good explanation what happened, right? And so we have to really dig into the data. At that time, believe me, we were under very high pressure, to find out what we believe something must have gone wrong. And then, but at that time, it's our first time to do the RNAseq analysis. We didn't have much experience, so we were quite worried. And just a little bit of statistic what we found in our sample, and we noticed about 5 to 10% of the fragment have multiple alignment. By multiple alignment, I mean that a read can be aligned to multiple locations of the genome by TopHat. Okay, and then of course you can see that would give you some ambiguity, right? Because it can only come from one location, right? And but when TopHat report multiple location, you need to know. You want to know which one is the correct one, right? But this number varies, right? Because it depends on what the parameter you set, or how many mismatch you allow during the alignment, things like that. And we also noticed that about 5% of the reported gene by Cufflink corresponding to known non-functional pseudogene. Okay, because these are already people annotated those pseudogenes saying that they're not functional. So, but yeah, a lot of them are, but those I talked to my collaborator, and then those are very, very suspicious because we have a lot of pseudogene pop out in the list, but those actually those are a small percentage of those that my collaborator are very sure they're wrong. So, but then another 15% are unknown. So we don't know what they are, and we searched through all this public database, and nobody said anything about those. Okay, and of course, yes, there may be some novel things that people didn't know about, but we don't expect that 15% is quite high for that percentage. So basically, we said, well, we better make sure that we did this analysis right before we claim that we discover anything new or contradicting with what other people say. So we look at the examples. This is one of the Cufflinks transcript. We just plot this black curve is the pile up height of the read aligned to this location. This actually I didn't show the intron part. So this purple vertical line representing the axon junction. I just left out. I just left out the intron part. Okay. So for this gene, there are 7 exons. Okay. This is a gene. It's called COM3. So don't ask me what function of this gene, because I don't know the function of this gene. But this is one of the genes annotated in a symbol. And then you can see that this is the number of read, right, aligned to the location. You can see this is quite a lot of read aligned to this location, to this gene. And then, but this level is really go up and down or go up and down, right? Quite a lot. And this red bars here represent the brown bars here mismatches. So which means that if you see this, that means that this location, if you look at the read, all the read aligned here, and see whether the base is they have the base agree with the reference genome or not, right? If they do not agree, but all these read are consistent with each other, but they just disagree with the reference genome, and then maybe this read takes this alternative allele there, right? And then we just use this bar to indicate that. So you can see that if it's a random sequencing error, you shouldn't expect to see this high bar, right? Perhaps pretty low, you wouldn't see it right down to the bottom here. So once you see this high bar, you know that they can't be very likely due to sequencing error. So I would like to see, well, let's focus on this little region, right? You can see this. There's a very low red curve here. Actually, that's represent a set of about over 2,000 fragments that can also be aligned to somewhere else. This is not their unique alignment. Okay? So that's caught our eyes. So what happened, right? And then let's zoom in, right? Focus on this and see what happened. So I truncated this is what in that green box. Okay, I just zoom in. And then this is, you can see clearly this red curve. And this is another location on chromosome 6 of the mouse genome. And then you see that quite similar red curve there. It turned out to be this 2600 fragment can also be aligned to this region as well. Okay? And then again, this black curve represents the power of hive of the read can be aligned to this region. You can see that in addition to this this group of a small group of fragment, there's a lot more read that are aligned to this region uniquely, right? They have no other alignment. But this region is an annotated region. We don't know. We couldn't find anybody said anything about this region. But if you look at this height, right, this coverage, it's as high as over 2,000 here. So it's not a small number that you would say, well, this is just random error or something, right? You want to ignore it, but no one can ignore this because it's too many fragments here. But what you can compare in comparison with this, you can see that whatever the value here, right, corresponding to the peak here, right? There's a reasonable hypothesis that could it be possible that all of those read actually came from there? Originated from there, but for one reason or the other, aligned here, right? And then actually verify that that's the case in this particular example. And in this case, actually, it is the actual axon junction caused the problem. Because in this region, there's no axon junction. And if you run TopHat before, right? So the way they recognize axon junction is there's some heuristic. They basically chop in the read into maybe 4 different pieces, and then hopefully some of these 4 different pieces can identify, have a good match, and then they sort of try to narrow down where the junction is. In this process, and then they could well miss that. If they miss that, then they couldn't get the right alignment. Another reason is that most of these alignment algorithms, right, if they often report the best alignment. based on the score, number of mismatches in India. And then if there's a second best with just a slightly worse score, they don't report that. So, which means it could be like the alignment here is just slightly better than alignment there. But then only this alignment is in your result. You never know it could be aligned to there. So we think that could be the problem. There's other reasons too. For example, if they have an unknown SNP for India, right? And but which make all the read come from here, and actually it's be more similar to the region here, because we should always keep in mind the reference genome we use is not the genome of the sample in the experiment. There will be always difference, and they're not sequencing error, right? Some of the difference you know about, some of them you don't, right? So those difference, unknown difference, may cause those problems too. But they're unknown, and then if they fail to map that, then it's very hard to rescue that. So in this example, we believe this, none of this read should be here. But based on this many read line here, no matter what algorithm you use to call this transcript, it will report this as a transcript, because it's just way too many to say that's not true. So when we did this analysis and investigation, exploratory investigation, we did compare between biological replica, technical replica to figure out what went wrong. And then we found that this pattern is highly consistent between all those replicas. And but if you have a different mouse strain, right, and then you observe similar things, but maybe that pattern show up for a different region or different pseudogene, and then, but it will always there. And this would cause a huge problem when we do differential expression analysis, right? Because when we, hopefully using different mouse strain to see how they respond, for example, differently to environmental factor or to drug, and because of this artifact of the alignment, right, we can mistakenly think that some gene or pseudogene is differentially expressed, but actually they're not. It's just the alignment give you the wrong impression. So, and because they're highly consistent between this, but across the comparison group they could be different. So, and when people study, okay, try different alignment algorithm, try different inference algorithm, when they thought, okay, they give similar result, maybe these are real, but actually these are not real. We also tried a different alignment algorithm, different inference algorithm, and then it's pretty consistent, but which means they all made a mistake at the same place. So now we basically focus our study on multiple alignment. Why? Because we think that's give us some indication where the problem might be. Okay? Even though at the beginning, the standard recommendation from the Illumina was that once you align your read, and then you throw out all this read with multiple alignment, because we're not sure where they came from, therefore they're not reliable. We don't want them to mess up with the inference. And then you only use those ones with unique alignment. That's a sort of standard recommendation that at least I heard at that time. That's also what we did at the beginning, but we realized that maybe it's too soon to throw those out. Maybe those multiple alignment give us information, like help us to crack this, because by having multiple alignment, those regions are similar, right? They might have this problem, and then we can use that information to help us to crack this, identify and crack this. So let's look at what are the main reasons for having multiple alignment. So first is what we found is a process pseudogene. So for process pseudogene, they don't have intron. Okay, so you think about alignment, especially if we read across two exon, right? And then if there's another location without the intron, right? Of course, it's easier to align, right? It's not surprising that they will have a better alignment score sometime, and so that's one of the reasons. Sometimes it's a non-processed pseudogene. It does have some intron, but maybe they have like equally, because they have similar sequence to the parent gene, and then So it's as a result, some of the reads still have multiple alignment. And the third reason was like simply like, we just have a repetitive sequence there. And then that's, would cause some of the read can be aligned to multiple places. So in that case, so how does an alignment algorithm handle all of this? And the reality actually is that we cannot really control what happened, right? So because all the alignment works like, okay, you have all this read, you give a reference genome, and then you specify how many mismatch indel you want to allow. And then the alignment algorithm basically try to find the best match, right? And output that. If there's more than one match with equally good score, all of this can be output, okay? That's basically what it does, which means that it depends on the, what the actual sequence and what the scenario, and where the read lined to the genome. So it could be one of this scenario. The first one is that, well, if we are lucky, then we can properly identify the splicing junction, and then maybe we can get it right. And then the next scenario would be that some of the read can only be aligned to the process pseudogen because it had given us slightly better score there. And then the third scenario would be it aligned to both locations with equal score. So both locations outputted. And then that's exactly what we observed in the previous example. You can see a small percentage, about 10% of the fragment have can be aligned to both. And then the other 90% are all unique. Okay, so this makes us believe that we really shouldn't discard all the read with multiple alignment. We should take advantage of them instead. So now our job becomes like, can we build a model to predict, right, which the transcript output by cufflink is actually corresponding to expressed gene, which one corresponding actually not expressed gene. So we want to build a binary classification model to help us to predict that. We try to avoid to use the term like predict gene versus pseudogene because pseudogene have its own meaning, and some of pseudogene are actually expressed. So we don't want to go into that area, like try to argue which one is gene or pseudogene. We basically just try to predict which one is expressed, which one is not expressed. So, and in order to avoid confusion, we don't call like an output from a cufflink a gene or a transcript. We just call them fragment attractor. Basically, they are the region that has a lot of read aligned to. Okay, so what we do is that we first run the standard pipeline, okay, and keep all the multiple alignment, and then Cufflink give us a list of those transcripts they reported, right? And then we take each of them, call each of them a fragment attractor. The reason why we want to focus on those, right, is A, it will help us to remove some of the noise or random alignment, and B, at least they are the ones that we need to be careful because the Cufflink already reported. We want to make sure which one is false, right? Which one is not. Right? If the Cufflink doesn't report it, then that's a secondary consideration. Okay, so from the Cufflink result, and we have a collection of fragment attractor. The first thing to do is to build a graph to represent the sharing of those alignment. Okay, so what we do is that we have a node to represent each fragment attractor. And we have an edge connecting them if there's a multiple alignment, right? If there's some read can be aligned to this location and that location, then we have an edge. And where the weight of the edge will be the number of reads that can be aligned to both. And then just for our visual effect, so we just basically color code them, but actually our model doesn't look at the color at all, right? So, and we don't use this information in the prediction model. This is just for our human inspection. So if we go back to this example, right, this actually turned out to be an easy case because this graph only have 2 nodes. We know that this one is a known, ensemble gene, and then this one is a known region, and then this is how many regions there. So this is actually a simple case, and sometimes we see it like slightly more complicated. And those are the, gene, and those are the annotated pseudogene, right? There's some sharing between them. And then sometimes it's like this. This is about 6,000 fragments. All have some kind of sharing, crazy sharing between them. And it's represent about 20% of the output from the cufflink. You can see that sometimes the problem can be quite messy. So now what do we do after we create the sharing? So this is like a cartoon representation. Let's see if they only have 3 of those. And then they have some kind of edge connect them. And this is a typical scenario. Like fragment attractor A is they have a similar sequence, right, to this part. And this means like there's a different exons. Okay. And then there's some rate can be aligned to both places, right? And then there's some rate can only be aligned to one of these 2 places. And that's true, you may have like a 3-way, sharing, right? And then there's this blue part means that, this is unique to C, not shared by others. And this is unique to B, not shared by others. Okay, let's see how we deal with this scenario. So what we do is that because if we look at the real data, we don't know the ground truth. It's very hard for us to build accurate prediction model. We start with a simulated data, okay? Because it turned out to be in this case, if you think the problem is with like, misalignment, right? And then we can simulate lots of read and see where they align to, right? And use that information to help us to build a prediction model. So if there's a shared alignment, that means there's similar sequence, underlying sequence. And then we want to look at what do we call consistent mismatch. That's corresponding to this kind of a vertical bars I explained to you. It's like this set of read takes the alternative allele, okay? And then, so number of those consistent mismatch, they're unlikely to be caused by sequencing error. It turned to be that if it's non-functional pseudogene, tend to accumulate, higher rate of those kind of mutations. So we should see more of those peaks than the functional peak. And then the number of exons, and then there are some other features that we use. I remember we have 14 features, but those are the interesting, ones. And then we build a classification model using simulated data. We can simulate lots of data, and then to do the prediction, because we know the ground truth, we know which one is correct. So in order to create the training instance, we always look at a pair of linked fragment attractor because they have shared alignment. Okay, so we first treat one of them as target. We try to predict whether this one is expressed or not. Right, using the information of the features of this one together with a linked fragment attractor. This one we call the assistant one. Okay, we use both features to predict this one, and then let's create one training instance, and then we basically want to see whether this one is unexpressed or not, based on all the information. And then we can just swap the row between them and predict whether this one is unexpressed or not. So for each pair, each edge, we have 2 training instances. And then typically, we would have, like, if I have a fragment A, we would have, several of them connect to that. So actually what we did was, this is simplest way of doing this. May not be the best way, but it works. So this is a target we try to predict. And then we have, because there are 4 edges, we create one at a time. Okay? And then we have 4 separate predictions. And then, let's see, 3 of them said probably expressed, and 1 of them said it's not expressed. And then our meta classifier said, well, if any one of them suggesting not expressed, we think this A is not expressed. The reason here is that if this A is not expressed, it means all the read aligned to this came from somewhere else. As long as you identify one of this other location that may be the origin of this read, you're fine, right? It may not come from all of this. It only comes from one. So that's why as long as one of suggesting this one is not real, right? Then we're fine. So after the prediction, the next step what we do is that we want to rescue those missed alignment. We want to basically literally go into the band file to take this pile of alignment to move it to the right location. We fix the band file in that way. Okay? And then after we fix the band file, now we come up with a new band file. And then we, this is the whole pipeline. We basically run Cufflink again to see what's the difference before and after this correction. And this is the standard pipeline. This is what we add. So basically, we create shared graph, and then extract features, doing the prediction, and then run the assembler again. Okay? And compare the before and after. So to show some of the result, how this pipeline works. So we have this, this in our study. It's F1 samples from a 3-mouse strain. It's a 6-week course. Just one of our goals is to study parent of origin effect. So each one of them, each sex have 3 to 6 replicas for each cross, and we use brain tissue. It's about, in this study, it's about 10 million fragment. It's a 100 base pair paired end read per sample. And to minimize the bias of these different mouse strains, because they have different distance, genetic distance to the mouse reference strain. So we actually created what we call synthetic genome for each of these strains by added the reference. Reference is very close to the strain black 6. And then to basically go to the non-SNP database or indel database to edit the reference genome by inserting those SNPs and the indels to make that as closer to the parent as possible. So to minimize the bias. So that example I show you is even after that. So still have that thing. So because this is a real data, so we don't know the ground truth. But what I can show is that this is the before and after. The blue dot are before, and the red dot is after. So this shows the number of reported transcript by Cufflink. It decreased. Okay? So, and each of this is one sample. We just group them by the cross and by the male and female, their sex. Okay? Which is good news to some extent. But we don't know this is suggesting good or bad, right? So it just say, oh, we have a less number of transcript to worry about. Now we say, well, let's compare with the reported transcript to the ensemble database. Because in the study, we didn't use any of the annotation from the ensemble, right? And then let's see, well, how are they corresponding to the known database? It turned out to be, despite that the number of transcripts dropped, actually, if we count how many transcripts exactly match the transcript in ensemble, the number actually increased. So this gives us some indication, well, maybe whatever we did is beneficial, right? So it's because we didn't use ensemble information during the whole pipeline. It was not part of the information we incorporate, but, you know, if that's true for every sample we have, that must mean something. It's like the direction of correction we make actually does help us to rescue some of the transcript. And then this is the number of pseudogenes reported. Okay? And then you can see that's dropped 2/3 of them are gone. There's still 1/3 remaining, because when we did that, correction was very conservative. If we're not 100% sure or 99.99% sure, we're not going to do any correction. And then this is the number of transcripts that are unknown, right, in the result. It's also dropped by about 20%. Okay, so for this set of results, we think, well, maybe this is quite good. So And then so it's quite interesting. And my collaborator really like, and after this one, actually, we run the differential expression again, and none of the top ones are no longer pseudogenes. So are the interesting real genes that suggesting so. So we also just to test our accuracy. This is a simulated data, so we can know the ground truth. So we also test a different alignment algorithm. Here I just show two different alignments. One is top hat, which I used before, and one is MapSplice is another one. Really, focus on identify. They're really good at identify axon junction. Okay, so we want to know whether that's the one that would make a difference or not. And then this is after what we call the gene seizure pipeline, and then before and after. You can see that the recall is pretty high, and then but the precision is not as high if you look at that. This is mainly because of cufflinks. If you have experience with cufflinks, they tend to over-report transcript or genes. They call it genes. So if you try to establish correspondence with the genes reported by cufflinks with the genes annotated in ensemble, and then you have several of them corresponding to the same gene, actually. You can find that. And in that case, when we calculate the precision, we always like, if let's say 3 of the genes reported gene correspond to 1 here, and then we count 1 of them is correct, the other 2 are spurious, because that's not the ideal result we want. It's maybe too harsh in some extent, but we just, because we always report many more genes than actual numbers, so we didn't like that part. So we said, let's just count those things or not right. Okay. So there are a few things we learned from this study. So of course, the existing pipeline is not perfect, and then it's useful to understand how it works. And then, as I said at the beginning, we learned this in the hard way, because it's always like we try to save time and just take whatever out there and then try it, and then it didn't work. We have to go into it and figure out what the problem is. And then throughout that process, we really learned a lot. And then also another thing is that the misalignment of the read can create actually bigger impacts than people originally thought. Because people say, well, that's maybe just due to random errors, and then which means all those misalignment should be uniformly distributed across the genome, and which means easy to overcome. But the reality is that actually they're very clustered and can really give you spurious reading of the transcript. And then the last lesson we learned is that we cannot really afford to throw away the multiple alignment, because they are the only hope we can use to correct this kind of mistake. So if you want to try this, you can download. It's open source. And that's the paper we published in last year's SMB. So since then, what we're working on now, because throughout this process, we also talk to the team who developed Top Hat, right? Because right after we published that, they also released Top Hat 2, which also aligned to the transcript. And then they said that, well, maybe, because the transcriptome is becoming more and more complete, even though it's still incomplete, but then we should rely on, take advantage of that more and more. So that makes us to think, well, maybe we should really use that in a more profound way. So in the previous study we have that we spent really a lot of effort, because for each sample we have, it takes many hours to just do the alignment. And then you take another couple of hours to the top hat take many hours, and the coupling take maybe 2 hours per sample. And then it took us many hours just to maybe change one parameter or figure out what went wrong. And so it's many hours on computer cluster, to be actually invested for that study. We thought that it would be nice if we can make it faster. So this is our current thing. So it's going to be presented in this year's SMB. So what we developed is really, we said, well, maybe if we can take advantage of the current known transcriptome, and then without doing the alignment, so maybe we can get it done faster. So our approach, what we did is that we studied transcriptome, and the extractor is extract informative, a small number of informative k-mers, and use that. And once you have the count of those k-mers in your read, without doing alignment, just count how many those occurrence, and you can use that to build build a big graph, not as big as Cufflink. It's a relatively small graph actually, and then do the inference. And then it turned out to be you can get as accurate result as Cufflink, is not more accurate. Actually, in our study, it's slightly more accurate if when you use simulated data. For the real data, we don't know which one is more accurate, but those two results have very high correlation. So if you believe one, you would believe the other. They'll essentially give you quite the same result. But if you look at the time spent, because again, when we did this with Rush, we only have implementations that work on one thread. For all these alternative algorithms, they can run on multiple threads, right? So for the rest, we all run 8 threads altogether. And then for we call the RNA scheme, we run 1. And these are top hat plus Cufflink, bow tie, so you can see all these things. And but if you compute this the CPU time per sample, this is about slightly less than about 10 minutes, right, per sample. And compared to all of this, you can see it's a lot. These are like hours. This is just 10 minutes, and then you can see how many times we can speed it up. This is 44 million, 100 base pair. So that's our new thing. And right now we're working on the final manuscript and also prepare for the release of the software. It's going to be released in the summer. We're also at the same time saying that, well, because this is only using the known transcriptome, right? Because which means we do not identify novel transcript. If we want to identify novel transcript, actually we can start from this, use this as a right anchor, and then to see what's not there, but also in your read as a way. But we're working on that. Don't have a result yet. But if you're interested, we can talk more. So this is just a new result we have. Thank you.
Yes.
How do you manage all these little parameters that have to be adjusted? This is a really good problem. Is there an optimization? You guys should be
well, one of the reasons we wanted to make it faster is that it's really very hard to try different parameters when each one takes many hours.
Right.
We spend so many, because one thing, well, maybe the number of mismatches we set the parameter, right? Allowed mismatches is not appropriate, right? And then we try different parameters, then, a few days passed, and then it's very tedious. So, but that's actually one of the motivation we want to use this, and then we can just use, set that to search for the best parameter setting. We have kind of a great computing-based network to really search through the parameter space. We can do that because each one is like no more than 10 minutes, sometimes faster. So, if you can budget like a day or two, right, yeah, then you can really search through our, reasonable parameter space to get you the best.
More questions?
Cuff merge and cuff diff to see if you can re-estimate the read abundances, if any of the spurious transcripts can be removed via their own method?
We tried. It cannot.
Did it
because
improve?
It's if the alignment is in the band file is wrong, the alignment, right? And then you can use IGV to just visualize that to see where they say, oh yeah, it's a big pile there, right? So of course, when you run those compare cuff diff, right, it will tell you They're different, right? And so we think that the only way to fix it is actually to go back, right, before you run Cufflink, and then, or in that case we run it twice, right? One is to help us identify where are the pile of read aligned to, right? And the second time was that, well, then we try to fix all this alignment and then run it again. So that's literally what we.
I thought that's one of the procedures of CuffDiff is when it converts the GTF back to the same file and then re-estimates the per transcript read.
So, but I don't think they changed the BAM file.
No, they go from the GTF back to the BAM file to convert to re-estimate the reads per transcript.
Yeah, but
given the comparison.
but you have to change the BAM file in order to get it right this time. Because if the BAM file doesn't change, you still get whatever you get, because that pile of read is still there, right? You cannot ignore. If that's real, the true alignment, then you cannot ignore, right? Because that's too many read there. But if you believe that all of them all of the read originate from another location, right, on the genome, you should really just edit your BAM file and change that. So that's what we did, yeah.
Any more questions?
So very impressive work. So I think these problems are somewhat recognized to many people, but it's really good to see some software that actually does the more systematic way to correct for this possible misalignment problem. And because a standard pipeline, actually, most of the large scale groups are making their own way to fix these problems. Mm-hmm. So it's not completely sure to how those probably more ad hoc method, but with some correction compared to, for example, Gervais group has 1,000 genomes, the RNA sequencing data, and with their own way, and they made their own way to do the quantification. Mm-hmm. So compared to those like state of the art way to do this quantification, so that's more complicated way than just using the standard pipeline that was recommended by Illumina. So do you have a sense that how much your outcome will be better than those kind of outcomes?
I think it really depends, right? Because we at that time we didn't have time to really compare all possible ways, and then the reason practical reason for us to suggest this way is that for other than the a handful of groups who can afford to have their own analysis pipeline, many people using the standard one, right? Yeah. And then they probably already did their top hat plus cufflink analysis, right? And having this is really easy for them to use.
Yeah, right? I totally agree that this is very useful. I just was wondering, well, how much this? I mean, if you might be able to show that this is actually better than the Gervais quantification, for example. So if that's something I think it'll be good to try and see.
Yeah, I agree. That would be good to try. But without trying, I'm saying that if they really intentionally try to identify this wrong alignment and correct for that, and I think it's maybe they can they can do similar result. But if they just account for some random error, right? Then we're dealing with different type of error, because these are not caused by sequencing error. These are to some extent not even completely caused by the difference between your sample versus reference genome. It's really inherent to the repetitiveness in the genome, so that once you get this wrong, and then if you don't correct that, because they're not random, they're very clustered, the problem. So.
Can I ask one more question? So, you showed the example in the mouse data where actually then the Castinia strain and the spread of strain is quite different from the reference strain. Right. So I would guess that in this case, mapping is much harder, and it's going to be very challenging. So your software will be very, very helpful. In the case of human RNA sequencing alignment, do you have a sense how much improvement you'd expect?
We haven't tried on humans, so I don't really know but I hope that it will give you some, improvement. But we haven't never tried. Yeah.
Thanks.
Hi, yes, great talk. So in the beginning, you mentioned some statistics like 15% reports like were annotated, or some like pseudo genes somehow may be wrong, or maybe have two copies, one pseudo gene, another one may be expressed. So those actually are bigger numbers, and maybe some like hidden scientific findings can be found from there. So I'm wondering, from what you do, or maybe your future direction, can you use your approach or improve it somehow to identify more things from the RNA-seq data? Like for example, maybe those some 15% annotated genes, or maybe something really you can find some new genes which were not predicted to be gene, or some genes which were reported to be genes but never been found in any study.
Well, we hope, but it's going to be a long process, right? Because it will take a lot of effort to really validate that, right? We can always make a hypothesis, but it's the validation part is actually would take a lot of effort. And then I actually rely on my collaborator to help me to work on that.
Okay, more question?
What can you do with longer reads? I mean, this is for short reads, where you have a lot of assembly, but with the nanopores, you'll have longer reads. So you should be able to imagine how you can use that even to improve this.
Right. So one of the recent projects we started is to really look at the PEG bio sequencing that have much longer read, but higher error rate.
Yeah.
We are currently developing algorithm that actually we want to be able to combine these two type of read, but we don't have my student is still implementing it, so we don't know any result. Don't have any result yet.
But
yeah.
There are several companies that have the nanopore sequences that'll actually lower the error rates significantly. So that should really help.
Yeah. How much difference in terms of the alignment? So we can see the percentage of read that are being able to align increase, I think, 7% on average. You see some of the difference in the. I forgot the number for how many transcripts are different, with a different depth. There are some difference. Yeah, it's not things that we can easily ignore. We tried that. That's why we use the synthetic genome, because it does create a difference.
More questions? Okay, so if we don't have more questions, so let's thank Wei again for the wonderful talk.
Thank you.